So hi everyone. We'll be continuing talking about the rest of ChIP-seq and then cover more of ATAC-seq for this lab. And still several announcements. I think I saw homework 2 TAs. They said please submit both HTML and or RMD for homework submissions. And if you're missing your HTML, remember to submit it before, I think, this Thursday. And then there will be a 10% deduction. But if you do not submit it, it will be like 0 points. So remember to do this before Thursday. Again, homework is due on next Monday at midnight. Check Slack if you have any question. And I really encourage you to come to our office hour on Friday and Saturday, especially if you have very individual-based questions, okay? And some clarifications on homework 3. I think these are like major comments. The first thing is for undergraduates, you do not need to run Q2 because MACS can directly take BAM files in Q3. And then for, let me turn off my Bluetooth really quick. Okay. And then for Q2, please use the provided to BAM files, but not the .fastq file. I know like in the question, it somehow mentions the .fastq file, but you can just ignore that. And then I think this is from a student. So she said like, we can see mouse TFs enriched as well. Like for example, FOXA2, like why is it the case? And I think she finally reached out to the author of SysTromDB, and it turns out that sometimes these mouse TFs might have better like reserved motifs or functions. So it's totally fine to use it, to use those motif name as well. And thank you very much for the student who reached out to SysTromDB author. And then for most downstream questions, whenever they said differential AR chip picks, they mention they mean Q4 output because your Q3 output is tumor alone and normal alone, right? And they might contain noisy signals, while Q4 output is like already filter out those in normal, but only keep those in tumors, the peaks in tumors. So whenever they mention differential, they probably mean Q4, okay? Okay, so I'll go with today's material. Here's a recap of the ChIP-seq analysis pipeline that we mentioned last time. And we already kind of did part 1 to 6 in your homework 3, part 1 to 4, right? And today we're going to talk more about how to integrate ChIP-seq with gene expression data and do more of functional enrichment analysis. So let's first talk about gene regulation model. So on the left, it's a gene transcription without any TF binding. You know, we have our RNA polymerase 2 bind to the promoter region, and then it just starts normal transcription. While when we have a TF binding, it might bind to the promoter region, which might help to recruit RNA polymerase, or it might actually prevent RNA polymerase from binding to the promoter. And you can also have TFs binding to the enhancer region that's like much further away from your TSS transcriptional starting site. And then those TFs will recruit other TFs binding in promoter that can then either increase or decrease RNA polymerase binding, which will then further affect downstream gene transcription. And usually for promoter binding, it's very close region, around 100 to 1 kb long before the TSS. While for enhancer, it could be much further distance and usually will form this loop-like structure when they are working in gene transcription, okay? And we also keep mentioning about regulatory potential scores because if you think about this model, right, how do I know if a gene is maybe affected by the TF from my ChIP-seq result? Or how do I know which TF might be actually affecting some downstream genes, right? And that's why we want to have this RP score. So I think Shirley somehow mentioned it in lecture, and in their lab's recent publication, they talk about a list of different factors that might affect TF regulatory potential. And this is like the most basic model. So if let's take a look at the equation, right? Just ignore the sum sign for now. So the delta is like distance to TSS. It could be any distance set by user themselves. For example, if you're more interested in the promoter region, promoter TF, you can set it to maybe 1 kb, 2 kb, 10 kb, right? If you're interested in more distant interactions, maybe you can set it to like 100 kb, right? And xijk means the distance of your peak to TSS of a gene. As x increase, we can imagine it's like an exponential decay, right, on both sides. So that's why in lecture Shirley said it's like an exponential decay of your RP score as you go further from TSS. And this is only for one peak I, one TFI on one gene J. You can imagine you can maybe choose to sum up these scores for each gene. Then you will know which gene might be affected more based on the ChIP-seq result. Or you can sum it up for each TF, then you will know which TF might be the ones that are like affecting a lot for downstream regulatory process, right? And this is like the most basic distance-based model. And in your homework, we already provide you this regulatory potential file in Q14, I believe. And it's calculated by a tool called Beta, also a tool developed by Shirley's lab in 2013. So if you're interested in learning more about this topic, maybe take a look at these two papers. I think they are quite nice. Okay, and then we want to talk more about BAT files and BAT tools because we use a lot of BAT format in this homework. So the very basic format of BAT file is the first 3 columns. You must have chromosome, the start of your region on that chromosome, and the end of your region on that chromosome. It's not limited to ChIP-seq, which is like peak region. It might also be gene region, for example, where the gene starts, where the gene ends. And okay, so I think that's for BAT file format. And in this homework, we ask you to use BAT tools to analyze those BAT files because it's developed in 2010, very well maintained. I also use it a lot in my daily practice. Just continue to the topic, not limited to ChIP-seq. For example, if I'm doing variant calling, I find some variants of human genome, right? I might be interested in, are they in the intron region or are they in the exon region, so on. And then I can use BAT tools to, maybe find the overlap of my variant's position and the intron region, okay? So I think the easiest way of checking a command tool is always, just type this tool's name in terminal. And on this figure, you will see like there are a list of different sub options that I didn't list all of them. But just for our homework, I think we're going to use subtract and window. But as I just mentioned, there are a lot of other very commonly used operations such as overlap. And I like how BAT tools, they actually give you this illustration to tell you how each subcommand work. They will also give you like small examples, like what will be in file A, what will be in file B, and what will remain after your command, right? So if you ever get confused about BAT tools, definitely take a look at their documentation just by searching the name of the subfunction, okay? So now let's talk more about the homework questions. So in question 12, we are asking you to determine what might be some AR target genes. So just back to our discussion of gene regulation, since this is AR ChIP-seq data, right? We're interested in learning about the genes that might be affected by AR regulatory regulations. And we specifically ask you to, if the peak is within 2 kb plus minus of the TSS, then you set it as a putative AR target gene. And this TSS information, where can we get it, right? it's within this refgene.txt file. I'll quickly go through it here. I'll show it again later, like what's within refgene. So this column, it's actually the refseq ID. And this is like the chromosome, the strand, and where this gene start, where this gene ends. And this is like the official gene symbol for the gene. But as you can imagine, this is quite different. I mean, there are some similar components, but it's very different from the BAT format that we just mentioned, right? And in order to use BATtools, you have to rewrite it to at least contain the first 3 columns. Otherwise, it will just not work. BATtools will just not work. So that's why we ask you to use Python to rewrite refgene.txt into the BAT format. And so I guess I can give a live demo here. Let me quickly, can you guys see my Jupyter Notebook page?
Yes.
Yeah. Oh, great, great. Okay. So here, the first line, we're just reading in the refgene.txt, right? And let's take a look of, by the way, Jupyter Notebook is something you can interactively see your Python output. Just like in R, when you click on something, you can see the output. Oh, sorry, let me import the packages first. Okay, so let's take a look at what's within TSS. Column 1, as we just mentioned, refseq ID, column 2, chromosome 1, and then strand, start, position of the gene, and position of the gene. And column 12 is like official name of that gene, all right? And so for this two, we are just reformatting refgene files into the BAT format that we just mentioned. Otherwise, BATtools cannot use it. And then we ask you to also in the homework to adjust start and end positions by strand, because if the strand is negative strand, it means you should change the end of that gene as the start of that gene. Okay, I hope this part is clear. So that's why you need to separate them into positive and negative strand. And then you need to rewrite, if it's negative, you need to rewrite its end as its start. And you might be wondering, like, what are we doing by +1 here? So if you think back about the RP model, and also the gene regulation, we only care about TSS, right? We don't even care about end position of a gene. But just in order to use BATtool, you need to have an end column. So that's why we are +1 here, just to create an end column for BATtools, okay? And then you just write your output to a new BAT file. So that's for, sorry, question 12. Sorry. We can also take a look at the TSS new that we finally create, right? So you will see that now it's in BAT format. We have chromosome, we have start and position. We also have additional like strand and also official gene symbol. You can also create more columns if you like, okay? So that's for the Python part. Let me continue. Okay, and then after you get this BAT file, we then recommend you to use BATtools window, which can help you to check if file, I remember it's if file B is within file A's distance. And for the window size, you just set it to our required window size, right? And then put your TSS BAT file here, and then put your differential pigs BAT file in file B. Oh, sorry, I might remember file A and B wrong, but you can take a look at their documentation. Yeah, actually, why don't I take a look at it right now? So a lot of times the file A and B sequence, the order matters, because you will have different outputs if you change file A and file B. So definitely take a look at their documentation. And as I said, they also have small example there, so you can know what will be remain after your command, okay? Let me go back. Okay, yeah, cool. So, and in the end, we ask you like, how many genes are there, right? You can use wc/l to count like how many lines are there in your output BAT file. So I think that's for question 12. I'll stop recording here. So now let's talk more about question 13 to 16 about functional enrichment analysis. So because, from question 12, we get a list of gene names, right? That might be affected by AR. But why does that matter? Because in the end, essentially, we want to know what's being changed in a diseased sample. And here, the left figure, for example, in leukemia A cell types, you can see these are like either the goal concepts or the functional pathway that's being enriched. For example, you can see apoptosis, you can see RNA processing, tube development, and so on. So, just from these enriched pathways and concepts, we can then actually know what's the biological mechanism that's being changed, right? Otherwise, just getting a list of gene names, we cannot relate it with the actual biological mechanism. And here is another figure. So it shows us, for example, you might be particularly interested in one pathway, and then you can take a look at how each gene, like how much each gene contribute to that pathway or to that enriched concept. But of course, we're not doing to this extent in our homework while using David. But I'm just suggesting like what could be some downstream analysis and why are they important here, right? So now let's talk about Q13. It's asking you to use David to run the AR target genes alone that you get from Q12. And you can also choose to combine it with the differential expression genes. That's like, maybe from RNA-seq result that are genes that are actually upregulated in prostate cancer compared to normal samples. And and then, take a screenshot of the enriched GO terms and pathways. I guess I can do a quick demo of, oh, sorry. So before going to David demo, you can read in this, because these files, they have multiple columns, right? They're not only the gene names. You probably need to read it in R or in Python, and then, find the intersection of the gene names, and then, output it to a file that only contains genes names, because David only takes gene names, right? So now I'll give a live demo of David. Can you guys see David?
Yes.
Okay, thank you. Okay, cool. I guess, so, just click on start analysis. I think you probably also used this before, right? But I'm just going to quickly show it again. For choose file, just choose the gene names file that you get after processing them. I'll quickly. Okay, just by the way, I'm quickly choosing some gene names. It might not be the same for your homework. Just don't refer this as any of your homework result, okay? And also, I think I saw someone asking like, which gene names should we use? Because there's like RefSeq ID, start with NM, and also like official gene symbol, right? But just in the upregulated files, upregulated genes, there is only official gene symbol. So I think you have to use official gene symbol in this case, right? Official gene symbol, yeah. So please choose this option and then choose gene list, just submit the list. Let's wait maybe for 1 minute. Okay, the web page is slow now. No worries. I guess I can continue with question 14 because it's also about David, and then maybe go back and check David later. Okay, cool. So question 14, so now it's back to the regulatory potential that we just discussed. We already provide you the file that has calculated the regulatory potential score for each gene. And we want you to select the top 1,500 genes with highest RP score. Again, you probably need to read it in either R or Python, and then select the top 5,000 genes based on the RP score. I would recommend order. Take a look in R console about the documentations of these commands, and then you need to write only the gene names for David to work. Okay. So I think, and again, we ask you to do this top 5,000 genes alone, and also overlapping them with those differential express genes. So this is a way of integrating with RNA-seq result, right? So I guess let's take a look at David again. Okay, cool. Now it's getting some results. Just click on functional annotation tool. And for example, we asked you to do Go analysis, right, which is gene ontology analysis. And there might be different concepts being enriched. I'll just randomly click some. Okay. Maybe you can just take a screenshot of like, not sure if you can see the small window that I'm open, since it's not on the same web page. But just for your homework, you can take a screenshot of this result page, and then maybe clicking some of these terms, and also maybe also clicking some of the pathways, and then take a screenshot. Okay. And just maybe take a look at their FDR and see if there's any difference for the same enriched concept. Okay. So I think that's for question 13 and 14, because both are asking you to, run David to find the enriched functional pathway or gene concepts, right? And then for question 15, so, it's asking you to compare the two methods. The first method is the 2 KB method, while the second method is 100 KB with RP score. What do we mean by binary here? Because if it's within 2 KB, we set it as a target gene, right? While for the regulatory potential, it's like calculating genes that's within 100 KB distance for that TF. For that gene, calculate the 100 KB TFs within that region, and then, summarize all the scores. So, it's like a free response again, and just compare, as I said, compare the p-values of some pathways, and then give us your thoughts here, okay? And then for question 16, so for what you did in 12 to 15, Shirley's lab actually has recently built a tool that can, perform like integrated and do all the things for you. So if you will first assign adjusted RP score to each peak, and then you will perform pathway enrichment analysis. So if you think about it, it's exactly what we did in previous question. So I can do a quick demo here. Okay. Oh, sorry. No, that's like wrong page. Okay, let me do a quick demo here. I think it's very straightforward. You can just upload your differential pick file, and then upload the differential upregulated gene names here, and then just click submit, and that's all. And then you will get a list of enriched pathways and Go analysis result. Maybe take a screenshot of those pathways, and you can click in it, but just a screenshot of those pathway names are fine. Okay. So I guess I'll go back. Yeah, so essentially it's just letting you know, like, now they have a more integrated tool that can do all the previous steps by a simple web page based tool. And I think that's for all questions for part 5. So now let's go through ATAC-seq and the last part of your homework here. I think Shirley already kind of talked about ATAC-seq in today's lecture. And ATAC-seq is an experiment tool to measure the open chromatin region, of our chromatin. So, our chromatin, our DNA is wrapped by histone protein, and maybe there will be some epigenetic modification that makes our DNA and histone pack either tighter or maybe more open. And if they're more open, we can use TN5 transposes to insert PCR primers or some specific sequence that you want to insert there in those open regions. And then, we can do PCR amplifications of these reads. On the right figure, it's a demonstration of how ataxic peaks would look like. So as we just mentioned, if some chromatin region is open, then you will see reads aligned there, right? And you will see peak-like signals again. And to be honest, we don't know any information about TFs there. I mean, if it's open, it's more likely to bind to TF, but like no one knows what's the actual TF binding there, right? Well, compared to ChIP-seq, we're directly pulling down a specific protein and see what's the DNA around it. So these two are like very different types of experiments. And there are some other measurements of chromatin accessibility you might heard about, like DNA-seq, 4C and Hi-C and 3C and etc. I think Shirley mentioned DNA-seq is not as good as ataxic, so most people switch off using ataxic now. You will hear more about Hi-C, I believe, in the next few weeks. It's just some other ways of measuring chromatin accessibility and maybe at chromatin scale. Okay. And here's like an overall pipeline of ataxic analysis. Just like any kind of sequencing data, we get the raw FASTQ file from sequencing companies, and you need to do some QC check there. You need to align it. For ChIP-seq, we use BWA. For ataxic, you can also use BWA or other aligners, and then do QC again. And then we can do peak calling using, actually funnily, we can use MACS here even for ataxic peak calling, because they also follow the Poisson distribution. So you can still model these peaks by that mathematical model. But all these previous step is already done by our side. So when you're starting with the homework questions, you already have the bed files, right? So all this pre-analysis and core analysis, it's like done. And in your question 18, we're asking you to do like peak differential analysis, like downstream analysis after you have the peak regions, the bed files. And in question 19, we ask you to integrate it with ChIP-seq analysis. So here is just showing what's the pipeline and workflow would look like. Okay, so now let's talk more about the homework questions. So in question 18, we ask you to use bed tools to find the peaks that are unique in tumor but not in normal. So if you still remember the functions I mentioned, there is a subfunction called subtract, which can help you to find the difference between two bed files. And we feel that since you already somehow used bed tools in previous question, maybe you can feel more confident of looking the documentation and then try to write your own command here. But again, if you have some difficulty, let us know in Slack, okay? And here we are piping. So this small vertical line is called pipe. We're piping your bed tool output to a new command. And this command will cut the first 3 columns. Otherwise, some downstream part might give you some complaint. So, but this part just means we're cutting the first 3 columns of your output from bed files, okay? And then this is like redirect the output of this command to your output for question 18, okay? And then after you get this bed file, we're asking you to run Systrom Toolkit again. If you still remember it from last week lab, it contains a lot of public ChIP-seq data, and it can help you to find, once you provide it a bed file, it can help you to find what are some other public data sets that has overlapping regions. So you will know a list of TFs that's like enriched from this experiment, okay, without looking at motif. And considering this is ataxic, you will know which TFs might be binding to those open regions or DNA, okay? And then in question 19, so now just take the top 10K ataxic peaks by fold change. I think, sorry, I cannot exactly recall which file, but I think it's probably in the file that we provide to you. It already contains the score, like, and also the fold change in the original BAT file that we are providing to you. So again, you can just, reading this table in R and then choose the top 10K picks, and then run Systrom Toolkit again. And if you remember, the output of Systrom Toolkit would be some dynamic figure, look like this, and then just compare it with question 18, like which method might give you more meaningful results, right? Question 18 is asking you to find the differential peaks in tumor versus normal, while this one is tumor only. So, like maybe give us your thoughts here. It's a free response again, okay? Okay, and here is like the very last question of our homework. It's back to ChIP-seq again, no longer relevant with ataxic. And let's talk more about LISA2 here. So actually, I was about to explain how LISA2 works, but I think Shirley already mentioned the overall process at the very last part of today's lecture. So maybe just watch her videos if you're very interested in how LISA2 is working. But essentially, we're just giving LISA2 a differential gene list. And in the end, it will return us what are some enriched transcriptional factors after the analysis, right? And just to let you know, the LISA2 is built based on public available ChIP-seq and DNA-seq data sets. And for both type of data, they're still modeling their RP as the exponential decay model we mentioned before, okay? So, but anyway, your input will be a list of genes, and the output they give you will be a list of TFs that are think to be important and enriched for these genes, based on these genes, okay? So here is question 20, and please remember to use this exact environment. Please do not install your own LISA2, but really use this pre-installed LISA2. And then, I mean, we can also take a look at LISA2's documentation later. But for LISA2, you can just input like HG38 is like our reference genome here, and put your upregulated gene list. Again, it's a list of the names only. And then, just they will give you output of a list of transcriptional factors. Just take a screenshot of the final output file content, like those maybe top 10 TFs and their scores. And if you're interested in, you can maybe try to simply search some literature about prostate cancer, because this gene list is upregulated genes in prostate cancer, right? And maybe you can search some prostate cancer literature and see what are some previously experimentally validated TF that are important, and see if they match LISA2's output, okay? So that's about LISA2, and I think that's like the last question of our homework. We still have a lot of time, so I'll be answering questions since then. Let me stop recording. Sorry. And in the end, don't forget about the 2 bonus questions. If you have time still, we encourage you to work on this. And okay, I think that's all for today. Feel free to leave, but if you have any question, I'll be here till around